In this bacterial physiology, we will discuss about the bacterial nutrition, bacterial requirement, how bacteria divide and grow, and about the bacterial growth curve. What is bacteria? Bacteria is a small or microscopic unicellular prokaryotic organism. We can observe the bacteria only under microscope, and it is measured in micron or micrometer. And it is prokaryotic organism, that means which lacks true nucleus. And there are so many types of bacteria in different size are available in this world, but the medically important bacterial size is about zero point 0.2 to 1.5 diameter, and length is about 2 to 5 micron or micrometer in length. Coming to the bacterial nutrients, the bacterial nutrient or bacterial nutrition is classified into macronutrients and micronutrients. Depending upon the requirement of the bacteria, it is classified into macro and micro. The macro means large, micro means small. That means the macronutrients bacteria require very large quantity that will help in the cell structure and metabolism of bacteria. And in micro, that require very small or tiny quantity of micronutrients for the bacterial functioning and also the enzymatic system of bacteria. The optimum nutrients for bacteria are water, source of carbon and nitrogen, inorganic salt, growth factor, and environmental factor. Water. Water is the most important requirement for the bacteria. Mainly, it is the principal constituent of cell. That means about 80% of the bacterial total weight is due to the presence of water. And also we can see that the integral part of protoplasm is a colloidal system. And this integral part of this protoplasm or cytoplasm is filled with this water. And it is a vehicle for entry and exit. That means the vehicle for the entry of nutrient into the bacteria, and also the waste materials will be comes out from the bacteria through this water. And also this water will have very important role in the metabolic reaction of bacteria. Based upon the carbon, nitrogen, and energy source, the bacteria is divided into autotrophs, heterotrophs, autotrophs, and chemotrophs. The autotroph, they will synthesize all organic compound themselves, but in heterotroph, they are unable to synthesize their own organic compound. And this phototroph, they will get energy from sunlight. That is the source of energy is from sunlight. And the chemotroph are they will require they will get the energy from the chemicals. Inorganic salt and other growth factors. Inorganic salt is also required for the proper growth for the bacteria, mainly the osmotic regulation and the enzyme system of bacteria require inorganic salt. The inorganic salts are anions and cations. The anions, they require phosphate and sulfate. As cations, they require sodium, potassium, magnesium, manganese, iron, and calcium. The growth factors are essential for the growth for the bacteria, essential for the growth of the bacteria. The vitamins, purine, pyrimidines, and amino acids are the growth factors required for the bacteria for their proper function. Environmental factors. Gaseous requirement. Based on oxygen requirement, the bacteria is divided into aerobic bacteria, anaerobic bacteria, and microaerobic bacteria. The aerobic bacteria means those organisms they require oxygen for their growth and multiplication. And aerobic bacteria is again subdivided into obligate aerobic bacteria. For example, obligate aerobic bacteria is the Mycobacterium tuberculosis. Is an obligate aerobic bacteria. In obligate aerobic bacteria, they will grow only in the presence of oxygen. If they are exposed to uh, oxygen-free environment, immediately they will die. In anaerobic bacteria is those organisms, they will live in the absence of oxygen. It's called as anaerobic bacteria. They will cultivate it anaerobically. And this anaerobic bacteria is again divided into obligate and facultative anaerobic bacteria. The obligate anaerobic bacteria means they will live only in the absence of oxygen. The examples are Actinomyces, Clostridium, Fusobacterium, Streptococcus are the examples for obligate anaerobic bacteria. They will be cultivated in the absence of oxygen environment. And in anaerobic bacteria, the one classification is the facultative anaerobic bacteria. The facultative anaerobic bacteria means they will grow in the absence of oxygen and also in the presence of oxygen. And most of the uh, medically important organisms are facultative anaerobic bacteria. Examples are E. coli, Streptococcus, Staphylococcus, Pseudomonas, Klebsiella. There are facultative anaerobic bacteria that can live in the absence and also the presence of oxygen. In microaerobic bacteria means those bacteria they will require micro or little amount of oxygen for their growth and multiplication. The example for microaerobic bacteria are Campylobacter jejuni and Helicobacter pylori. And next about the capnophilic organism or capnophilic bacteria are they require carbon dioxide, about 5 to 10% of carbon dioxide for their growth and multiplication. The examples are Brucella botrys, Neisseria species, and Pneumococci require a small amount of carbon dioxide for their growth and multiplication. Temperature requirement and pH. Based upon the temperature requirement, the bacteria is divided into psychrophilic bacteria, mesophilic bacteria, and thermophilic bacteria. The psychrophilic bacteria grow below 20 degrees Celsius. Mesophilic bacteria grow best at 25 to 40 degrees Celsius. And this thermophilic organism will grow at 45 to 80 degrees Celsius. And most of the pathogenic organism or medically important pathogenic organism will come under this mesophilic organism because the optimum temperature of medically important bacteria is 37 degrees Celsius and the optimum pH of medically important bacteria is 7 to 2 to 7 to 6 is the optimum pH of medically important bacteria. The optimum means during this optimum condition, the bacteria can grow well. Or we can also say that in optimum condition is the condition where the bacterial growth and multiplication will take place at higher level. Bacterial division or bacterial cell division. The bacteria multiply by simple binary fission. That means one bacteria will be divided into two daughter cells. So this is the bacteria. The bacteria, the outermost, we have the bacterial cell wall. Inner to the bacterial cell wall, we have plasma membrane. And inner to the plasma membrane, we have the bacterial, this is genetic material or circular DNA is there. And all the organelles can be seen inside this bacterial plasma membrane. Then this is the mother cell. This mother cell will attain this maximum size. That means the bacteria will reach its critical mass. And after that, the next step we can see that this is the duplication of chromosome or the duplication of the genetic material. That is the circular double-stranded DNA. And this circular double-stranded DNA will be divided into two identical complementary strands here. And after that, here you can see this is the septa. The transverse septa will be formed here, and this transverse septa will be divide the bacteria, this mother bacterial cell into two daughter cells. And this is the bacterial cell division, and this is the mother cell. This mother cell will attain its size maximum, and after that, the duplication of the genetic material, that is the double-stranded DNA molecule, will start divide into two, and the septa formation can be seen here. And this transverse septa will divide this mother cell into two daughter cells. And the generation time means the time required for bacteria to give rise into two daughter cells. That we will call it as a generation time. And also we can say that the time between two cell division is called as generation time. And depending upon the organism, the generation time will be different. For example, in the case of E. coli, Escherichia coli, the generation time is about 15 minutes, and the Staphylococcus will be 13 minutes. And like that, depending upon the organism, the generation time will be different. And about the Mycobacterium tuberculosis, the Mycobacterium tuberculosis generation time is 15 hours. That means depending upon the organism, the generation time will be different. The generation time is the time required for bacteria to give rise to two daughter cell. We will call it as generation time. So how bacteria divide, that we already discussed. The first, the size of the bacteria will be increased, and after that the cell division take place, and two daughter cell will be formed. Two identical daughter cell will be formed by simple binary fission. There are two types of bacterial counts are there. One is the total count and viable count. Total count means the total number of bacteria present in a medium, we will call it as a total count. And the viable count means the total number of viable bacteria present in a particular medium. The viable means live bacteria, we will call it as a viable bacteria. That means in total count, we will count the total number of bacteria, whether it is dead or alive. We will count the total number of bacteria. And in viable count, we will count only the live bacteria. We will count the live number of the bacteria. Bacterial growth curve. When a bacterium is inoculated into suitable liquid medium and incubated at suitable temperature and pH, the culture passes through different phases of growth. When bacterial count of such culture determined at different interval and plotted in relation to time, a growth curve will be obtained. That we will call it as bacterial growth curve. So in bacterial growth curve, first we will inoculate the bacterium into a suitable liquid media, and we will wait for incubation. And during incubation, the bacterial count are made at interval after inoculation, and we will plot a graph in relation to time and number of bacteria, and we will get this bacterial growth curve. So this is the bacterial growth curve graphical representation, and this is x-axis. X-axis is for hours or time, and this y-axis for the viable count. Here we will can see the viable count and time. And bacterial growth curve is having mainly four phases. First one is the lag phase, log phase, stationary phase, and decline phase. In this lag phase, this is the first phase. Here you can see the number of viable count of the bacterium will become stable because just we have inoculated the bacterium to this culture media, that is liquid media, So what will happen? The bacteria have to adapt this particular environment, and during this lag phase, what will happen is the bacteria will produce or it will start synthesizing enzymes or other metabolics for the multiplication of bacteria. And just after this lag phase, if we can see, this is the log phase or exponential phase. During this phase, the bacterial growth will be exponentially will increase. And during this time, the number of bacterial multiplication will be maximum. So we can see this number of bacterial viable count will be increased exponentially. Just after to the exponential phase, this is the stationary phase. In this stationary phase, you can see the viable count will be stable because the number of newly dividing bacteria will be equal to the depth of that bacteria. That means in here you can see the viable count will be stable. There is no increasing in the viable count because during the stationary phase, what will happen? They will have the nutrient depletion because culture media will have the specific amount of nutrient or limited amount of nutrient will be in the culture media and bacteria will utilize this nutrient. So they will have the less or depletion of the nutrient. And also the depth of the bacteria will be equal to that of the newly formed bacteria. So in the stationary phase, the number of viable count will be stable. That means the depth of the bacteria will be equal to the newly formed bacteria. And after to the stationary phase, this is the decline phase. Here you can see the number of viable count will become declining, and the number of or the rate of newly formed bacteria will become less, and the death of the bacteria will become more. So the viable count is declining in this decline phase, or this is also called as death phase. And why it is declining means because already you will have the nutrients. The culture medial nutrient will be already utilized by the bacteria. When the bacteria rate will become more, more nutrient is required. So already this bacterial, we are giving a limited number or limited amount of nutrient. So that nutrient depletion will be occur. So bacteria will not get proper nutrient for their multiplication. And also during this phase, there will have the bacterial accumulation of toxins will be more in this culture medium, and due to the toxins and also due to the autolysis of the bacteria, the death rate will be more than that of the newly formed bacteria. So in decline phase, the viable count will become less or declined or reduced. Bacterial growth curve of total count. In bacterial growth curve of total count, here we will count the total number of bacteria. Total number of bacteria means the number of live and dead bacteria we will count here. In viable count, we will count only the viable number of bacteria. That means only we will count only the live bacteria. Here in total count, we will count both live and dead bacteria. So this is the lag phase. Lag phase will be same as that of the viable count. That means the number of bacterial cell will not increase. It will be stable here. And the second phase is the log phase. In the log phase, the bacterial total number of bacteria will become increase exponentially. And after this log phase, here you can see in this stationary phase and after this stationary phase, the graph will be continuously steady without any decline in the total number of bacteria. While in the case of viable count, the stationary phase will be stable, and due to the death of the bacteria, the viable count, the graph will decline. Here in total count, the graph will not decline here because we are counting both the death and also the live number of bacteria. This is about the bacterial growth curve of total count.
Subscribe to Simply Micromed. If you're curious about medical microbiology, infectious diseases, and unraveling the mysteries of tiny organisms, hit that subscribe button. Join our community of science enthusiasts, and let's explore the microscopic world together. Remember, knowledge is contagious. Spread it by liking and subscribing. Thank you.